Assalamu alaikum students. Welcome back to our O-level biology lecture series. In the chapter, we have already studied the main biological molecules, carbohydrates, lipids, proteins, and DNA. Today, we are going to study an important practical part of this chapter, which is very useful for examination. In this lecture, we will study how scientists test food for different nutritions. We will study the food test for starch, reducing sugar such as glucose, proteins, lipids. These experiments help scientists and students identify which biological molecules are present in a food sample. So let's start. Firstly, we will discuss the test for starch. Starch is a complex carbohydrate that is commonly found in food like potatoes, rice, and bread. To test for starch, we use iodine solution. Now discuss the procedure for this. Place a small amount of the food sample in a test tube or on a white tile. Add a few drops of iodine to the sample. Now please observe, if the starch is present, the iodine solution changes color from brown to blue-black, and if starch is absent, the iodine solutions remain brown. This iodine reacts, and this is because the iodine reacts with the structure of the starch molecule, producing the blue-black colors that confirms the presence of the starch. Now the second test is for reducing sugars. Reducing sugars includes simple sugars such as glucose and maltose. To test for reducing sugar, we use Benedict's solution. Now discuss the procedures for this. Place the food sample in the test tube. Add Benedict solution to the sample. Heat the test tube in a hot water bath for the few minutes. Now observe, if the reducing sugar is present, the color of Benedict solutions changes gradually from blue to green, yellow, orange, and then at last brick red. A brick red precipitate indicates a high amount of reducing sugar. If the solution remains blue, it means reducing sugars are not present. Now the third test is for proteins. Proteins can be detected using Biuret test. Now discuss the procedures. Place the food sample in the test tube. Add sodium hydroxide solution. Then add a few drops of copper sulfate solution. Now it starts observations. If proteins is present, the solution turns purple or violet. If proteins is absent, the solution remains blue. This is because the chemical reacts with the peptide bonds in the protein, producing the purple color. Now it's time for the test of lipids. Lipids such as fats and oil can be tested using ethanol emulsion test. Its procedure is place the food sample in a test tube, add ethanol and shake the mixture well. Then add water to the solution. If lipids are present, a milky white emulsion forms. If lipids are absent, the mixture remains clear. Now we discuss its reason. Lipids dissolve in ethanol but doesn't dissolve in water. When water is added, the lipids form tiny droplets that create the milky appearance. Now discuss the safety precautions. When performing food tests in the laboratory, it is important to follow safety rules. Wear safety goggles. Be careful when using hot water bath. Handle chemicals like iodine Benedict solution carefully because they may stain skin or clothes. Avoid testing or directly touching food samples used in experiments. Now we discuss the applications of food test or the importance of the food test. Food tests are useful in many areas of science and everyday life. They help us identify nutrients in different foods, study the chemical compositions of the food, understand how the nutrient supports the body functions. These tests are very important for biology practical examinations. Let's quickly review today's lesson. Iodine test is used to detect starch. Benedict test is used to detect reducing sugar. Biuret test is used to detect proteins. And the ethanol emulsion test is used to detect lipids. Each test procedures a specific color change that confirms the presence of a particular biological molecule. This brings us to an end of chapter 4, biological molecules. In this chapter, we learned about the proteins, carbohydrates, lipids, and DNA food test for different nutrients. Make sure to revise these concepts carefully because they are very important for all our biology exams. Thanks for joining these lectures. Allah Hafiz.